ये वीडियो सेल में होने वाले सेल साइकिल रेगुलेशन को समझाएगा। इसके साथ हम ये भी बताना चाहेंगे कि हमारी क्लासेस ऑफलाइन और ऑनलाइन दोनों मोड में चलती हैं।
The cell cycle is a tightly regulated process. Checkpoints ensure error-free division. Molecular signals greenlight each phase.
Inside every cell, a protein network acts as a microscopic control system, ensuring genetic information is passed on according to a strict, predictable schedule. The cell cycle functions as a unidirectional engine. It coordinates the expansion of tissues and the ongoing repair of damaged organs through a sequence of specific, regulated phases. In 1855, physician Rudolf Virchow codified this reality with the phrase omnis cellula e cellula, establishing that every living cell stems from a pre-existing cell. In 1882, Walther Fleming observed this transition in the dividing cells of salamander larvae. Using early aniline dyes and brass optics, Fleming tracked the physical separation of chromosomes, coining the term mitosis for the process. To understand how this works, we must map the specific regulators driving the process, specific cyclins, kinases, and genetic checkpoints. This division is an architectural process where each phase must be completed in a specific order. Any failure in this regulation allows the growth to proceed unchecked, leading to the development of a tumor. This matrix maps the chronological progression of the cell cycle through 4 distinct phases: G1, synthesis, G2, and mitosis. These empty slots represent Cyclin-Dependent Kinases, or CDKs, the central catalytic drivers of the cycle, remaining completely inactive on their own. In the early 1980s, scientist Tim Hunt studied dividing sea urchin embryos and identified proteins whose concentrations spiked and degraded with each phase. He named them cyclins. Identifying these oscillating proteins provided a unified model for cell cycle control across species, a contribution recognized with the 2001 Nobel Prize. When a cyclin binds to its target CDK, it forces a physical structural shift in the kinase, activating its ability to phosphorylate downstream proteins. This binary activation serves as the biochemical circuitry that advances the cell out of its current phase and into the next. The G1S border is the mammalian restriction point. Here, retinoblastoma protein, RB, clamps the E2F transcription factor, preventing premature synthesis. Upon receiving growth signals, cyclin D CDK4/6 adds phosphates to RB, breaking its grip. This frees E2F to trigger genes needed for DNA replication. To prevent accidental activation, the cell uses inhibitors. The INK4 family blocks specific kinases like CDK4, while the CYP-KIP family wraps around and halts entire active complexes. If the DNA strand fractures, sensor kinases ATM and ATR detect the break and stabilize the p53 protein, the guardian of the genome. If damage is minor, p53 activates the p21 gene, physically halting CDK complexes to allow DNA repair. But if damage is irreparable, p53 activates BAX and KUMA, driving the cell to dismantle itself through programmed cell death, or apoptosis. By continuously monitoring its DNA integrity, the cell makes a binary choice: fix the damage or initiate self-destruction. Why does the cell cycle only move forward, never reversing into a previous phase? The ubiquitin proteasome system ensures unidirectionality. Specific ligases tag phase-specific proteins with ubiquitin chains, signaling a shredder, the 26S proteasome, to dismantle them. This irreversible shredding governs the transition from metaphase to anaphase. It is managed by a large complex called APCC, pairing with cofactor CDC20 to target the inhibitor securin for destruction. Destroying securin releases the enzyme separase. Separase acts as a biological scissor, slicing through the cohesin rings that hold chromosome pairs together, allowing them to pull apart. By permanently destroying the proteins of the previous phase, the cell creates a biochemical ratchet that enforces forward momentum. If chromosomes are divided unevenly, the resulting cell faces massive genetic instability. To prevent this, the spindle assembly checkpoint monitors tension. Any chromosome that is not attached to a spindle fiber generates a protein signal that locks CDC20, freezing the cycle in place. While these mechanics are widespread, their execution varies. Budding yeast cells perform closed mitosis, where they separate chromosomes without dissolving their nuclear membrane. Mammalian cells perform open mitosis, dissolving the nuclear envelope entirely to give the spindle fibers access to the genetic material. Certain cells modify these rules even further. Megakaryocytes bypass division entirely, replicating their DNA up to 64 times to build the volume needed for platelet production. These examples show that while the core engine is conserved, evolution has engineered specialized bypasses for specific tissue functions. When these specific architectural breaks are damaged, the entire regulatory logic of the cell breaks down. Cancer results from the failure of these mechanisms. Mutated p53, broken RB locks, and hyperexpressed cyclin D allow the cell to bypass its internal restrictions and divide without limit. Clinical research identifies how persistent environmental toxins specifically target and mutate these tumor suppressor genes. Medical institutes track these molecular breakdowns to understand the origins of malignancy. Even in healthy anatomy, local environments drive different cycle speeds. Intestinal stem cells divide continuously, relying on specific signaling pathways to manage the constant replacement of the gut lining. The stability of any tissue depends on the balance between growth signals and the internal checkpoints that protect the genome. Cell cycle regulation is the system that manages the tension between tissue growth and genetic stability. Mastering its mechanics allows us to define precisely how the biological engine maintains itself over time.